talked about different ways our DNA can accumulate damage. But now we're going to talk about the measures that are in place to correct those damages. So by now you know that those ways that we can damage our DNA can come from inside and outside of the cell, so are induced and spontaneous. And it's important to note that our DNA is the only molecule that a cell would rather repair than replace. And think about it. So, our DNA is precious. We only have one set of chromosomes in each cell. It's not like proteins where, oh, this is damaged, destroy it and make another one. We can't just destroy our DNA and start from scratch. We have to be able to repair the one that is there. If not, the entire cell would have to die. So most of these mechanisms that we're going to talk about act on the fact that the DNA is a double-stranded molecule and that it's complementary, because if you know one strand, you can tell what the other one is. So if you have to repair it, if you take away an area on one strand, you just use the other one as a template and rebuild it. I'll also note that there's a lot of redundancy in these repair mechanisms. So there are several repair mechanisms that can fix one type of damage. So like when we talk about the thymine dimers, you'll see that there are a couple different repair mechanisms that can recognize it and fix it. So here is a list and a description of the repair mechanisms that we'll talk about. And now we're going to go into detail a little bit about how they're done. And the thing that you should notice is what's similar in these things. So most of them are composed of one or more proteins, okay? They're multi-step processes. So first, you got to detect the problem. Then you got to remove the problem, and then you have to repair that DNA. And when you repair that DNA, we're using replication mechanisms like DNA polymerase and ligase. So the first one we'll talk about is direct repair. This is just going in and reversing whatever's going on. So from the prior video, you've seen that UV light can cause these thymine dimers to form. Well, this photolysase comes in and just cuts those bonds between the thymine dimer. Now the thymine is able to bond again to adenine, okay? And this is going to be called photoreactivation, and it requires light, okay? And then this is found in bacteria, fungi, most plants, and some animals. And think about why is it important that most plants have this system? Well, where do you find plants? Hanging out outside, and they need sunlight to do what they do. So they have to have a mechanism in place to repair any damage caused by that sunlight. Another mechanism is called base excision repair, BER. So this targets non-bulky DNA damage. So this is like methylation, deamination, things that happen to individual bases. So how this works is you go in, you take that base out, and then you're going to cut around it. This is initiated by DNA glycosylases. So those DNA glycosylases recognize and remove the base, okay? So now you're going to have an apurinic or apyrimidic region where you just have that base taken out. Then you have this AP endonuclease that comes in and cleaves that phosphodiester bond. Then we're going to take that sugar out, okay? So now we have an empty space. Well, DNA polymerase comes along, puts the correct nucleotide in, and it's going to use the other strand as a template to figure out what that correct nucleotide is. DNA ligase comes in and seals that backbone, and now our DNA is restored to normal. We have nucleotide excision repair. This is similar to base excision repair, except for now this targets bulky and we're excising whole nucleotides out of the strand. And again, the intact strand is our template. This is found in all eukaryotes and prokaryotes. This uses proteins called these UVRs, okay? And there are a bunch of them here listed, and then it's going to use DNA polymerase and ligase. So the steps that we're going to talk about have been outlined in E. coli. It's going to be similar in eukaryotes, but as most things we see, eukaryotes, it's a little more involved, and they have more proteins. So nucleotide excision repair. What's the first step? Recognize we have damage. So we have a complex here of UVR A and B that tracks along the DNA looking for damage. Here it has identified a thymine dimer, the second mechanism we've talked about for repairing thymine dimers. So after it finds it, the UVA goes away, and now UVB is sitting there, and UVRC comes. So now you have a complex with the C and the B. Well, the C is going to go ahead and cut on either side of our thymine dimer. Then we have a D that comes in that has a helicase function, and it's going to allow us to remove that damaged region. Now we have this damaged region is gone, and the UVR proteins go away. Well, DNA polymerase comes in, uses the intact strand as a template, rebuilds the damaged strand, DNA ligase comes in, and now we have restored it back to normal. So we have mitchmatch repair systems. Okay, these are what we want to think they're a little less common, but in fact we know that this occurs quite often. I won't say often, but it appears in our DNA. The first thing we have is DNA polymerase itself has proofreading capabilities, so it is able to go and undo any mistakes that it might have introduced. And then if it misses it, well, we have a mitchmatch repair system that's able to come in and do the job as well. This system is found in all species, and it relies on DNA replication. So what do I mean by that? It's going to use the daughter strand during replication. It's going to repair the daughter strand on replication using the parent strand, and it knows which is which by DNA methylation. So the parent strand is still fully methylated, whereas the daughter strand, which was newly synthesized, is not. So this is going to use these MUT, like mutation proteins. So in our mismatch repair, and I have two pictures that just show the same thing. This one actually shows those MUT proteins. So the S is going to slide along the DNA looking for a mismatch. When it finds it, the S and the L form a complex that binds to the H, which is already there on our hemimethylated sequence, which is on the daughter strand. H is going to make a cut in the non-methylated strand, which is the daughter. U is going to separate that DNA at the cleavage site. We have an exonuclease that comes in and digests it. And now we have an open area where DNA polymerase can come in and fill in the gaps using the intact parent strand as a template, and then ligase comes in and seals it up, and our DNA is restored to normal again. Okay? Another type of repair mechanism we have are for our double-stranded breaks. Now, we talked about double-stranded breaks when we talked about our chromosomal abnormalities and how if a double-stranded break is not put back correctly, you get deletions, inversions, and translocations. So that should sound familiar to you. Now, naturally, in a human cell, there are about 10 to 100 breaks per cell per day. This can be caused by those ionizing radiation, chemical mutagens, some chemotherapy drugs, but it's also caused by those reactive oxygen species that are a normal byproduct of cellular metabolism. Okay? This double-stranded breaks is breaking entire chromosomes. So these are the most dangerous types that we'll talk about because we can lose out on several different genes when these occur. So we have homologous recombination. Now, we talked about homologous recombination when we talked about recombination, and this is a way that we can repair DNA damage, and this relies on us having our homologous chromosomes nearby, okay? So remember those holiday junctions and all of those things? Well, this is the other model, the double-stranded break repair mechanism. So this is going to have a D loop, but what happens is you have the DNA that has the damage. You're going to cut around that damage. Then you're going to use the homologous chromosome that's okay to invade that strand and serve as a template so that you can fill in the gaps. The other flavor of this is non-homologous end joining. So now you don't have to have the sisters, okay? But what happens here is you have these proteins that recognize that there have been double-stranded breaks. And so they come in and they want to protect those ends. So these end-binding proteins want to protect the ends. And then they're going to recruit other proteins that's going to come in and help us. And what we want to do here, though, is connect these broken chromosomes. But first, they're going to start to digest away some of the areas to leave these overhangs. And then DNA polymerase can come in and fill in the gaps, and DNA ligase can come in and seal our backbone. But I will note here, so the first thing is that we don't need a sister, okay? So we don't have to rely on homology. So this can happen at any stage. But with the caveat that we can introduce small deletions. So because we are not looking at an intact chromosome here to help us, we don't know if this is the full strand. So we could very well be missing an area of chromosome here, okay? So that's important to note that although this might seem a little easier so than homologous because we can do this at any time, the downside is we may very well lose small amounts of genetic material. We have a translesion synthesis in place. Now, this requires these translesion replicating polymerases. What are those? Those are polymerases that can replicate through the damage, okay? Like, you don't have to repair it. I can do this still. But these tend to be more error-prone replication. And now, so when we talked about DNA polymerases in replication, these are those translesion replicating polymerases. But they're not as faithful as the regular polymerases. So that means they can keep introducing more errors, okay? So although we can do it, they're not as faithful. So we talked about how these mechanisms are in place. Well, I would note a couple things. One is over time, because we use them so much, they're going to get exhausted, and this could be a hallmark of aging. Number two, if they are damaged by a genetic mutation, this tends to lead to diseases in humans, specifically cancer, okay? So they have been, this nucleotide excision repair has been linked to skin cancer, and mismatch repair mechanism have been linked to a type of colon cancer. So having defective DNA repair mechanisms can lead to disorders, diseases, and ultimately cancer, depending on which mechanisms are defective. Okay, so I hope this was informative, and I hope that you took away big picture items. Okay, so I know you hated those multi-answers so much, but this is the same idea. It's like, well, what can you recognize that are in common here? Don't get so bogged down by all the tiny, minute details. Like, what's the big picture? Okay.